And we've come full circle with our Challenges in Nanomedicine session. We're back to FDA. And we have a talk by Dr. Katherine Tyner from the CDER office, talk on regulatory consideration for drug products containing nanomaterials. Thank you.
Thank you very much for the invitation to speak today. As mentioned, I'm going to be talking about some of the challenges that we see on the regulatory standpoint for drug products containing nanomaterials. And I recognize I'm standing between you all and lunch, so I will try to be brief. So we saw a disclaimer in the beginning. We'll see one at the end, too. What I'm saying is my personal opinion, and I am not endorsing products. All right, quick overview for the Food and Drug Administration. The FDA regulates foods and drugs, as well as medical devices, tobacco products, dietary supplements, and a whole host of other things. And just something to keep in mind, all these different products have their own regulatory and legal authority surrounding them. So I am talking under the Center for Drug Evaluation and Research, and our center, we call CDER, reviews applications for new and generic drugs, new indications for already approved products, and active ingredients and labeling for over-the-counter drugs. And we do this through a multidisciplinary review process where drugs are evaluated for safety, efficacy, and quality. And if you hear a CDER talk, you're going to hear those three words no matter what. So Anil briefly mentioned about this. The vast majority of products that we see coming across our desk are through different types of submissions. We have the investigational new drug applications, INDs, and that's the request to start a clinical trial, so your phase 1s, 2s, 3s. Once those are completed, you have your new drug applications. That's the approval for marketing on the US market. The BLAs, which are comparable to the NDAs, but for biologics, and then the abbreviated new drug application, which usually refers to the generic process. Okay, so let's talk about nano. Very important. Regulation and law do not separate nanotechnology products from products that do not contain nanomaterials. So they go through the same regulatory review process and standards that any other product would have. Fun fact, the FDA does not define nanotechnology, nanomedicine, or any related term. What we have are points to consider, which came out in an overarching agency guidance in 2014. There's 2 points to consider when we're looking at products that may involve the use of nanotechnology. One is whether or not the material has at least one dimension in the nanoscale, roughly 1 to 100 nanometers, or whether or not there's a material that neat properties can be attributed based on its size, and that can go up to 1,000 nanometers. So when I'm talking today about nano, I'm talking under that umbrella. All right, so Anil previewed this slide for you all. My group just recently finished a retrospective analysis of drug products that have been submitted to the United States since the 1970s, so almost 50 years. And if you're interested, so we collected a lot of data, we did a lot of analysis, and all of that has just recently been published online in Nature Nanotechnology. So I'm going to refer you all to the sites for the more detail, but I do want to give you some main highlights of this analysis. So since the 1970s, you see that we have been reviewing and approving drug products that contain nanomaterials. Our plenary speaker yesterday mentioned that nanotechnology has been around forever, before the nano buzz really took off. But you will see that starting in the 1990s, we really did start to get an increase of submissions for drug products containing nanomaterials, and all types, including generics as well. And this really seems to track well with the deliberate and intentional use of nanotechnology when nanotechnology became more popular and also maybe had more funding opportunities as well. So we've had roughly 400 drug product applications that contain nanomaterials. It's not a static number. People keep on submitting them, and we keep on reviewing them. And we have over 60 NDAs and approvals, so US marketing approval for these drug products containing nanomaterials. So overall, CDER uses a science-led and risk-based approach for the review of drug products containing nanomaterials, and I oversee an overarching CDER Nanotechnology Working Group, which is a multidisciplinary team that's comprised of experts across CDER, and we are working to identify risks to safety, quality, and efficacy, as well as identify areas of improvement in the review process based upon risk analysis. And there is a risk analysis in teeny tiny, oops, wrong one again. risk assessment in teeny tiny letters, and if you're interested, there's a citation there that goes over the nuts and bolts of how we actually did the risk assessment that looked at some of the major gaps and some of the major areas that we could work on for drug products containing nanoparticles. And what we found is that current regulatory framework and review processes can adequately manage the potential risk for drug products containing nanomaterials. But there are some areas that we can will benefit for additional work. And so I've highlighted some of the ones that would be considered challenges. I've underlined them. So one of the main things, physicochemical properties, such as particle size, can significantly impact the product performance and safety. And to measure them, you oftentimes need special analytical methods to characterize the nanomaterials. Now, as we went through this historical review of submissions that we've seen that throughout the years, and especially very recently, there's been an increase of the variety of nanomaterials we've seen, and importantly, an increase of complexity of those materials. And with that, we see a huge diversity of drug product applications. And so we're addressing all of these challenges through three ways: research, guidance, and outreach. And since research is near and dear to my heart, I'm going to spend most of the time talking about the research we've been doing. So the first research I'm talking about is more of the paper projects, the desk projects, looking at the data that we have and seeing what the trends are and what we're seeing. So what we've seen is a huge variety in terms of drug products containing nanomaterials and the different materials. So we have about a third of the materials that are liposomal drug products, and really the first generations, your nanocrystals, your iron colloids, do take up a significant chunk of what we have seen historically. But this slice of the pie is becoming interesting. It's getting a little bit bigger. It's pushing out some of the first generics, and it's also getting segmented. Lots more little strips where we're looking at the different types of materials that we're seeing being submitted to us. If we do the analysis and we look at indications, we see a similar breakdown. So cancer indications, so products that are targeting cancer indications, roughly about a third of the products we've seen. But then we have just an incredible diversity, really covering all the spans of indications. And we're seeing all routes of administration. So really, these products are being considered a formulation tool for a whole range of products. Okay, so we have this information, and we can take a look and say, okay, we have the breakdown of what we're seeing. So we can go ahead and look at the highest population, which are liposomal drug products, and we can dive deeper. And so this was a project that was done by one of my postdocs, Mamta Kapoor, where she pulled every submission that the FDA has seen for liposomal drug products and then documented all of the different interactions that the agency had with the sponsors. So the questions that we submitted to their sponsors and the response back, and then grouped all of that information and all of the kind of questions that we had and grouped them into different categories to see if we could find overarching challenges or areas where there was a lot of discussion. And we found two main challenges for liposomal drug products. One was that the identification and appropriate characterization of the critical quality attributes. And directly related to that was the suitable control strategies to control those critical quality attributes during manufacturing, storage, and delivery. So, challenges really circling around characterization, and that shouldn't be really all that surprising. That's always a fairly strong topic when a bunch of regulators get together and talk about challenges with drug products containing nanomaterials, the types of characterization and the novel techniques used. But we can go back to our analysis and look to see what type of characterization has been provided to us by the industry. And beyond a doubt, the most common techniques for drug products containing nanomaterials is you're dispersing the product and then you're shooting a laser at it. So either dynamic light scattering or laser diffraction. But throughout the years, again, looking at what we're seeing in the past, say, 5 years, we're seeing a really dramatic increase in the use of dynamic light scattering as well as high-resolution imaging, and that high-resolution imaging being also more complex, so coupling the imaging to different techniques. So again, we can take a look and see the wedge of the pie and see what we've been seeing in terms of the challenges that the regulators have seen over the years. So if we look at dynamic light scattering, what challenges has the FDA seen in drug submissions using dynamic light scattering? We've seen a lot of different challenges in terms of the validation. Having the instrument being used outside of range, having validation performed weirdly, for better words, or not standardized across the different products, If you're doing a fit for purpose, not using a standard reference material that is actually representative of the product they're looking at, and all sorts of different ways to prepare your sample. And so, so looping back to Anil's talk to kind of close the loop for that, a lot of this can be addressed by standardization. And so CDER has started to be very active in the development of both documentary standards as well as reference materials, so we can start closing the loop in terms of what we're seeing and provide a unified voice in terms of some of these very common techniques. And the reason we can do that is if we start at kind of the same starting place for industry and the regulatory authorities, where we understand what we're talking about, that ultimately reviews the number of review cycles, so the back and forth between us and the industry. The more of those you have, the longer it takes to get a drug on the market. So if we're reducing those cycles, we can speed this to the market, which ultimately is everyone's goal. Okay, so I hope I was able to explain that CDER's maintained a robust research profile in the area of drug products containing nanomaterials. And so thus far, I've really talked about the desk projects. So the projects where we're just sitting at the desk and just going through tons and tons of data, which I find lots of fun. But I hope you can understand how these projects can directly inform the laboratory projects. So we can take the work that we're doing by saying, we have this many products and we have this overarching challenge with this group of products. And it makes it a very strong case when we go up for review funding, saying, and these are now the lab-based projects that we want to work with. So we have both a laboratory and a non-laboratory component. We also have extramural funding, so we have grants with companies, with academic institutions, and we also have straight-up collaborations with anyone that wants to just work freely with us in the labs. Our research focuses on either product-specific, but more generally try to do class-specific projects or just general questions surrounding the nanomaterials and drug products. So we do a lot on the quality considerations, so the different ways of characterizing. We also have an active research group that looks at emerging analytical technologies. So seeing what's on the market, what's coming through, and then seeing, then we take the instrument and try to break it, see where it would fail when we're looking at drug products. Of course, have a pharm tox component as well as equivalents for complex drugs. And I'd like to point you all in the direction of two research or review articles that recently came out that go over the last 10 years of FDA CDER nanotechnology research. So if you're interested in seeing what we've been doing, those are the articles to go to. Okay, so we do research. We also get our opinions across, or current thinking across, through guidances. So we have the agency guidance, which I alluded to earlier in the talk. We also have class-specific guidances, such as a liposome guidance, as well as multiple product-specific guidances. One thing that's missing, but has been mentioned in the talk before, is this gap right here. Usually you have an agency guidance and a center guidance, and then go into the class-specific. We've been scheduled to have a center guidance for nanotechnology since 2015. I think 2017 is the year. Have high hopes. Just a note that when the guidance comes out, it's published publicly, and there's an availability for public comments. And I do encourage anyone that has a comment, either positive or negative, but we usually see the negatives, to go ahead and submit those comments. They will be read by me, as well as the working group, and they will be addressed. And it does help make these guidances stronger and more useful to the overall community. We also participate a lot in outreach, such as giving talks like this, but I also want to point your attention to a couple of things that have come out recently. One of the ones is a theme issue from the AAPS journal, and the hard link is here. It's co-edited by some people in CDER, and it's a topical collection of some of the current issues we've seen with drug products containing nanomaterials. It has industry perspective, academic perspective, as regulatory perspective. And it has a couple of papers talking about developmental considerations for liposomes, nanocrystals, modeling, and some other components as well. So it's a nice collection, everything in one website, just ready for you all to use. We also do a lot of conferences, and there's two that have recently come out that have white papers associated with them. I know that a lot of people have talked about the New York Academy of Sciences at this meeting already. And as Anil alluded to, we also are active participants now in a lot of the standard setting organizations. So working with ASTM, ISO, as well as USP to take the knowledge that we've been gained with what we've seen, what our challenges are, and hopefully address some of those challenges through standardization. And we also are recommending, based on our information, specific developmental of standards based upon our analytical techniques and what we're seeing in CDER coming to us. All right, so general considerations, both of them involve talking to the FDA and both involve characterization. So characterization plays a critical role in establishing the quality of the drug product. Oftentimes you need to have specialized analytical techniques to characterize that nanomaterial. In both cases, the recommendation is to come talk to us early so we can be on the same page. That again reduces that back and forth between the agency and industry and hopefully speeds along the whole process. And with that, I would like to acknowledge my wonderful team, as well as my colleagues at the FDA, and I'm happy to answer questions here or in the hallway.
Thank you very much, Katherine. Katherine, questions? Comments? Compliments?
Thanks, Frank.
Yes, please.
There's a lot of work around trying to standardise DLS and make it fit for purpose, but fundamentally it's not fit for purpose. So why don't you just ban it and tell people to do it properly?
So I'm sorry, I don't see where you are, but I will just talk to the global. Thank you very much. So every analytical technique that we see is going to have its pros and cons. And I agree that we can't be using analytical techniques past what their fit for purpose is. That said, there are some, the benefits that we see, we can't ignore that, and we also can't ignore how the number of submissions that were coming through. So if we're seeing a bunch of submissions coming through and they're all wrong, that's something that we want to address in some fashion.
Nearly all wrong.
Well,
But they're nearly all wrong, and they're nearly all with DLS, so there's a direct correlation. And it's a chicken and egg situation where the submitters think, well, that's what the FDA want.
Right.
And the FDA says, well, that's what the submitters are spinning. So it'd be quite good to break the cycle.
Right, and it's a good point in terms of the methodology and the characterization. And so one way that we can do it is for light scattering, all the light scattering techniques, saying, hey, this is what you can actually use it for. If you're going to use it within these parameters. And then to your second point of what FDA actually wants, we get that through the guidances. So we'd be able to hopefully through that explain, maybe not this for one area, but this for another.
There's a question there.
Thank you for your excellent overview of the activity, and I commend on the work because it's amazing. During this session this morning at the UNCL, a question was posed about the requirement for concentration characterization,
Right.
which is not being considered. Could you speculate on that?
So I haven't listed kind of we have done some work on what we typically ask for in terms of critical quality attributes. It's very case dependent. And so in terms of concentration, it's not something that we've asked all that much. It really depends on the product. But typically, I mean, the things that we have seen overarching is size distribution, and surface characterization.
Yes, please. Let's work it out.
Thank you very much for your really excellent talk. Very interesting topic. Do you have any emerging? Okay.
Go ahead.
Do you have any emerging analytical techniques for recognizing exogenous nanoparticles contaminating the product? Imagine a case. I have an exogenous material coming through a high-pressure homogenizer.
Right. Thank you for that question, because that's actually something that we are working on right now. So you have a particle in solution, and you need to know what that particle in solution is, but you also have to know if there's other particles in solution and what they are and if they are bad. And when we're starting to talk about the range where we're talking about nanoscale materials, that becomes a very challenging issue. So whereas I can't actually comment on specific techniques that we would recommend right now. We do have a robust area that we are looking at that. And so we'll take instruments into the lab and we'll look at different ways. And so we're looking at the fit for purpose, actually, to the gentleman's first point, to see what is good and what is not. And then if we find something that's usually pretty good, or if we find ones with limitations, we do publish that through peer-reviewed literature to kind of see where we are at. So we won't typically say in a guidance use this technique, but we will talk about the research that we've done surrounding it. But it is an active area. We don't have any recent papers on that right now. Mm-hmm. Thank you for that.
Last question, Sesha.
So you mentioned in one of your bullets equivalence of these complex products, and you talked but mostly on the analytical piece, but really how does it evolve from, even if you have great analytical techniques, how does it evolve, and how do you then use it to show equivalence, or just, to ultimately it's a clinical performance of the drugs? How do you relate to that?
Thank you for the question, and unfortunately our colleagues from the Office of Generic Drugs were not able to participate today. Currently there is pending legislation in the US Congress about generic drugs, and so we've been asked to hold all comments right now.
All right, with this I'd like to thank all the speakers of this session for their contributions, for their presentations, and I give the word to Beat for some announcements.